How do you turn off this signal? It's an important process to be able to turn the signal on as well as turn the signal off. So rhodopsin kinase and arrestin work together to phosphorylize that opsin molecule and then keep it in that phosphorylated state. The GTPase and cyclic GMP activated protein, that changes the amount of cyclic GMP located within the molecule. And remember, the amount of cyclic GMP is what regulates that sodium channel from opening and closing. So to review this process one more time, let's look at a photoreceptor cell, and let's look at how these changes occur during dark and light. So during the dark, there is a high amount of cyclic GMP, and therefore these cyclic GMP gated channels are open. Therefore, it is sustained, and they sometimes call this the dark current. And the dark current is going to be something that you have to remember. It is active during the dark. When you're exposed to light, that channel closes. Because remember, you have a decrease in cyclic GMP, which is gated to that sodium channel. So in the light, you get a hyperpolarization. Okay? So remember, dark versus light activates these photoreceptors in a slightly different manner. Now that we have talked through the process of rods, let's now switch to cones and different wavelengths of light. So rods are going to be responsive to just light, but not to different colors of light. Cones have the distinct responsibility of being able to sense color changes, and it does that by different frequencies of light. S cones are in the more blue-purple range, M cones are in the green range, and then L cones are in the red range. So these cones work in the same manner as the rods, except for they're excited by a little different frequency of light. So their goal isn't just to collect light, but is to distinguish between the colors. Signal convergence. How photoreceptor layers go through and converge is how you're going to get a complete picture. So here you can see that sometimes you have cones that are hooked to a single bipolar cell, which is then hooked to a ganglia cell, and then the ganglia will transmit its information eventually through the optic nerve. If you want to, though, have very, very high distinguishing power, such as with a certain color or an acuity, you will have these hooked up in a one-to-one relationship. Otherwise, they're hooked up in small photoreceptor fields. Spots in the retina will be different depending upon how they are arranged. Such as in the fovea, you might have a nice one-to-one relationship between cones and bipolar cells. In other parts, you might have a greater receptive field. But in general, rods have a large receptive field and cones have a small receptive field. Now, since you have 3 different neurons here in a row, we have to also eventually talk about how they are going to signal each other. But what we want to first talk through is how light flows versus data. Light flows from the top down towards the photoreceptor. Once the photoreceptor is activated, it sends data up. But it's good to always remember that with this process of having light and dark current responses, the cells are basically always sending signals back. In one form or another, they are always communicating. So the data is always flowing up towards the optic nerve and into the brain. On and off-centered responses. So you may wonder why or how you have these 3-stepped process response work, and how do you get, with only 3 cones and 1 rod, how do you get all the immense amount of colors and shapes and all the different things that you see through the eye? Part of the process works on and off-centered responses. So remember, photoreceptors are always releasing glutamate in the dark. This is your dark current. As you have this dark current response, it is always delivering you information that it is off. Remember that bipolar responses are graded, meaning that you can have a plethora of different levels of transmission. It does not necessarily just transmitted or not transmitted. Okay, here is where you're going to start getting a nice distinguished pattern of signal. You have some ganglia cells that are hooked to a photoreceptor that are on cells. These are on cells because they utilize an inhibitory metabolic glutamate receptor. These metabolic glutamate receptors, if glutamate is present, will turn off that particular cell. You have off-centered neurons that will be stimulated by glutamate. So even though you have a response that's occurring, you're seeing something, photoreceptors are sending information, some bipolar neurons will be turned on and some will be turned off. So you always have various neurons going on and off, all the way through the retina. This allows for very specific responses because there's enough overlap in these cells to be able to get you fine discrimination between objects.